Thank you, Arjit. Now, next speaker, we have Professor Yueh-Hsia. Uh, uh, Topic of the talk is the hidden impact of air pollution on digestive system. Study reveals link between air quality and intestinal health. Professor Lu, please.
Okay, thank you. Good afternoon, everyone. I'm delighted to have the opportunity to present my study today. So I just modify the title, just simplify it. So the title is the hidden impact of air pollution on the colon health. Okay, so this is the outline of my presentation. So first, I will introduce the health impact of air pollution, especially focus on the colon health, and then I will briefly share our finding from Kaohsiung PM 2.5. Okay, so accumulated evidence has revealed that the adverse effect of air pollution, including premature death, which is increased mainly mortality from stroke, heart disease, chronic lung disease, lung cancer, and acute respiratory infection. So a new satellite mission is developed by NASA and Italia's ASI. They will investigate the effects of particulate matter in air pollution on human health. So they aim to produce the unique map of particulate matter air pollution that epidemiologists will use to study how different types of particulate matter affect our health. So basically, they will use the space-based data to study the human health and improve the life. So the ambient particulate air pollution is the sixth leading risk factor and top environmental risk factor for worldwide deaths. So many studies reveal that the infection from bacteria, fungus, or virus such COVID-19 can be exacerbated by air pollution-related health problems, making people more susceptible to severe illness and adverse health consequences. So how can air pollution affect our health? PM in air pollution has been shown to increase our risk in many health problems, including the chronic lung disease, the lower respiratory infection, brain, heart disease, heart attack, lung cancer, and even the fetus. So this is the example, the paper published in the 2016. So the author demonstrated that circulating factors in response to air pollution can prime the brain microglia cell. And this review paper published in 2020. So they conclude that the exposure to air pollution is associated with many altered composition of gut microbiome, particularly in animal model. And IARC published a monograph in 2016 on the evaluation of the carcinogenic risk to human. So in lung cancers and urinary bladder cancers, the air pollution can increase the risk. And for the other cancer sites, the association were inconsistent. However, in recently published paper, for example, this paper, they conclude the study published in the late 30 Brazil. So they conclude the long-term exposure to air PM 2.5 increase the risk of mortality for many cancer types. And there is no threshold for the effects of PM 2.5 on the mortality for all cancer sites. So even low level PM 2.5 concentration had significant impact on cancer mortality. And this paper also showed that in the Thailand, they found that increase in black carbon, organic carbon, and dust PM 2.5 has linked higher risk for colon cancer. Okay. And this is the most recent, most newest paper. The author comprehensively examined the association of long-term air pollution exposure, air pollution-related DNA methylation, and the risk of the survival. So their findings suggest a significant association between air pollution and both the cancer, colorectal cancer incidence and survival. Okay, so in Taiwan, air pollution is also an important issue. So this is the real-time air quality index visual map during the winter season. So you may see that the air quality index in Kaohsiung is pretty high. So we cooperate with Professor Tang Huanglin from NCU and the NHRI group in Taiwan. We aim to clarify the underlying molecular mechanism of PM 2.5 impacts on the colorectal cancer. So first, we focus on the PAH in air pollution. PAH is a group of organic compounds consists of two or more fused aromatic rings, and most of the individual PAH has been classified by IARC as either probably carcinogenic to human or possibly carcinogenic to human. And it is important to differentiate between their structure variations to respond to this chemical contamination and understand their effect and transport in the environmental. So we can use the ratio. We can calculate the ratio of low molecular weight PAH to high molecular weight PAH to characterize or predict the origin of the PAH in the environment. So we compare the Kaohsiung PM 2.5 and the SRM. SRM is a fine particulate matter, and this is a classified reference material. So our finding, we found that the Kaohsiung PM 2.5 is may be the pyrogenic peril emission, material combustion, aluminum smelter emission, and non-traffic emission. And then we use the animal model and the in vitro model. So since the inhaled PM can reach the GI tract through the mucous ciliary transport, so we use the oral vaginal aspiration to stimulate this process. So the 24 weeks of exposure of Kaohsiung PM 2.5 in mice lead the reductions in the colon length and the significance increase the pancreatic weight. And the reduction in colon length suggests that the continuous exposure to Kaohsiung PM 2.5 induces a chronic inflammation. And the overall expression of Ki-67 positive signal, this is a proliferation marker in the chronic cell. The positive signal was significantly higher in the PM 2.5 exposed group than in the control group. And we collect the fecal stool from the mice and we want to identify the biomarkers of the gut microbiome after exposure to the PM 2.5. And we found that the Kaohsiung PM 2.5 can increase the proteobacteria and reduce the vermicutes and vertical microbiome in the stool. So this is the feature of this data indicate the mice microbiota dysbiosis, and this is associated with the intestinal inflammation. Okay, and then we further analyze the microbiome profile, the phylogenetic and the histogram display LDS score, and we calculate the difference in genus level abundance between the PM 2.5 and control exposure mice. And we did found several biomarkers increase in the Kaohsiung PM 2.5 exposed group. So the increase the proteobacteria indicate that the dysbiosis in the gut microbiota, and the patient with the inflammatory bowel disease has the same profile. They have lower gut microbiome diversity, more proteobacteria, and fewer vermicutes compared to the healthy individual. And then we use the cell model to identify the effect of the PM 2.5, and we found that the PM 2.5 and the SRM can induce the activation of inflammation and increase the inflammatory mediators, and this may exacerbate the tumorigenesis and progression of CRC, the colorectal cancer. And then we cooperate with Professor Lin Tang Huang, and we collect the patient from the Lansing Hospital. So we collect the patient with the colorectal inflammation, and Professor Lin provide us the this patient PM data estimate from the using the satellite remote sensing.
You have 1 minute, please.
Okay. Okay. So we found that the serum level of ILA are increased and correlate with the regional anthropogenic pollutants. Okay. So we published our result last year in this journal, and this is the schedule cartoon feature in our study. So due to the time strain, please feel free to contact me later if you are interested in our study. Okay, so this is the final implication of public health. So while the diet and genetic are known to affect the digestive disease, our study show that the air pollution may be a hidden risk for the gut health. So we highlight that the need for more research on environmental pollutant, and suggest that protecting air quality could be more critical for overall health than the previous thought. Okay, thank you.
Thank you very much, Lo, for very important one, biomarkers and air pollution.